carefully and slowly. All right. Okay, so that's all in the way of questions, and make sure you practice thinking about the anatomy that we talked about so far in that way, in that flow type of way, the way that we've been drawing it out kind of schematically. Although that's not how a heart looks, it's really helpful in, as far as thinking about left to right, you know, what valves are encountered, what vessels are encountered before we get back into the right atrium, and so on and so on. So you can get a lot of questions that just ask you purely about the flow or the sequence of flow from one side to the other, or from one vessel to the other. Okay, so let's get into our second objective here, which is to understand the electrical properties of the heart. And so we'll start off by looking at the conduction system of the heart. So how does electricity move through the heart? How does the heart create its own weight and rhythm? So there's this idea called autorhythmicity. And autorhythmicity is the ability to generate its own rhythm. And the heart has this. A few of the cells of the heart, which we call autorhythmic cells, have this property. They can spontaneously depolarize. And so because they can spontaneously depolarize, they basically set the rate or the rhythm because they're the first cells to begin that electrical activity. So autorhythmic cells also provide a pathway for the spread of this electrical activity throughout the heart. And so there are two types of autorhythmic cells. We've got pacemaker cells. These are the initial cells that have the fastest pace, and they're going to start the action potential, as I mentioned. So they're going to set the tone for the rate and rhythm of the heartbeat as we know it. And then the other type of autorhythmic cells are the conduction fibers. So they're not actually initiating action potentials as fast as the pacemaker cells, but they can, to some extent, spontaneously depolarize, but they're more important for passing that conduction through the heart very quickly. And we'll talk about examples of those fibers coming up in a bit. Now, these are only the autorhythmic cells. There are other types of cells in the heart. We call them contractile cells, and they make up a much larger proportion of the entire cardiac cells. So a very small percentage are these autorhythmic cells, which are pacemaker cells and the cells that make up conduction fibers, but then a larger chunk of the cells are really contractile cells. And so the pacemaker cells are really important for starting the rate, spontaneously depolarizing, setting the tone for how fast the heart is going, the heartbeat as we know it, whereas the other cells, the contractile cells, are important for force generation or for contractility of the muscle. And the pacemaker cells are not as good as generating force as the contractile cells are. Okay. So let's look a little bit more in detail at these cells. So the pacemaker cells, we said, can spontaneously depolarize. And that is, they can change the membrane potential to generate an action potential. So very similar to what we saw in terms of the events, the electrical events of a standard action potential. The only difference is there is no stimuli as such that makes this happen. Pacemaker cell can spontaneously play around with the gating mechanisms of channels, open channels and closed channels spontaneously. And that in and of itself, we know will lead to membrane potential change, which is what gets us to an action potential. They also help to coordinate and provide rhythm, or the heartbeat, as I keep mentioning. And so the pace at which the pacemaker cells initiate those action potentials eventually ends up being the heart rate or the heart rhythm. And I'll talk about a small exception with the SA node and how the autonomic nervous system kind of plays a role there. The second set of cells here, the conduction fibers, these help to rapidly transmit the action potential that is initiated by our pacemaker cells out throughout the myocardium. And again, although these are not the contractile cells, these cells are not really playing a role in force generation, but really in that conduction or that rapid transmission of the action potential. So they're not generating a rate or rhythm as fast as pacemaker cells. We'll talk about this. They're slightly slower, but again, they're not the slow contractile cells that are really important for force generation and not so much for the rate or rhythm of the heart. So the pacemaker cells in the heart are the SA node or the sinoatrial nodal cells. These are what set the main rate of the heart. So this is the primary pacemaker cell or primary group of pacemaker cells. This is located at the top of the right atrium. I'll point that out a few slides down. And then we've got a second group of pacemaker cells, which are the AV nodal or the atrioventricular nodal cells, another cluster of pacemaker cells that's located in between the left and right atria, really close to the tricuspid valve. And that's also going to be important in setting that tone, especially if we have problems with the SA node. And I'll talk about some examples in a bit. And then we've got the conduction fibers. So we've got the internodal pathways. These are the fibers that are in between both nodes. So in between the SA node and the AV node, there are fibers called the internodal pathway or internodal fibers. We've also got the bundle of His fibers, which are really located along the ventricular septum. So the ventricular septum is the wall that separates the left and right ventricles. And along that septum is the bundle of His fibers. And then we've got the Purkinje fibers, which are really located throughout the inferior parts of both of the ventricles. So we've got the internodal pathway or the internodal fibers, we've got the bundle of His fibers, and then we've got the Purkinje fibers, really at the bottom of the heart and more so located in the ventricles. And all of that will make a little bit more sense when we actually look at the propagation of that electrical activity. So the autorhythmic cells, we said, this is the different group of autorhythmic cells. And then on the right, we can see the firing rate for these cells at rest. So for the SA node, we've got a firing rate of 70 to 80 action potentials per minute. And we've got a little asterisk here because this is really influenced by the autonomic nervous system. In fact, scientists have proved that if you take an SA nodal cell from that cluster of SA nodal cells, if you take one cell and you remove it from the influence of our physiology, remove it from our body, put it into a petri dish or so, it will actually have a firing rate closer to 100 beats per minute. But because inside of our physiology, there's the influence of the autonomic nervous system, we talked about this, the fact that we've got not an equal contribution from both, but there's slightly more parasympathetic influence. And so that slightly predominant parasympathetic influence actually slows down the SA nodal firing rate to about 70 to 80 action potentials per minute. And that's where we get our resting heart rate of about 70 to 80 beats per minute in terms of our actual, in terms of that vital that we can measure on our cells. So it's really outside of the body, really about 100 beats per minute, but inside our physiology comes down to about 70 to 80. The AV node has a firing rate of about 40 to 60 action potentials per minute. The bundle of His, again, much slower, 20 to 40 action potentials per minute. And then the Purkinje fibers, likewise, much slower, about 20 to 40 action potentials per minute. So we can see the progressive slowing of the rate of action potential firing that happens from the SA and AV node, which are the main pacemaker cells of the heart, SA being much faster than the AV node, obviously, and then the Purkinje fibers and the bundle of His, which are the conduction fibers, having a slower action potential rate per minute. That is to say that they can still spontaneously contribute to the rate and rhythm, but they're not setting the rate and rhythm because they don't have the fastest rate. Now, cardiac cells are usually linked by gap junctions, and we'll talk about the junctions that separate one cardiac cell from another and some of the structures that are present there. But gap junctions is one of those structures, and it's really important because gap junctions are what help to propagate the action potential throughout the entire myocardium. So it's going to go from the pacemaker cells to the conduction fibers and then eventually make its way to all the contractile cells really quickly. And so gap junctions are going to facilitate the transmission of that electrical activity from one cell to another and eventually from different fibers all the way throughout to the contractile cells. And I mentioned this earlier, the fastest depolarizing cells are what set the rate. They're the ones that control the other cells. In this case, that happens to be the SA node. There can be SA nodal defects. So if you have maybe coronary artery blockage and you lose blood supply to one part of the heart, let's say you lose blood supply to your right coronary artery, which actually supplies the right side of the heart, you're actually going to lose blood supply to your SA node. And some people after a heart attack or after coronary artery blockages, their SA node is not working because it's kind of damaged from that loss of blood supply. And so the AV node has to take over, or we could insert an artificial pacemaker, which I'm sure many of you know about or have seen, you know, in the cadaver lab. We have a few cadavers that have pacemakers as well. So basically, if you don't have a working SA node, the AV node can take over. It's much slower, but the ideal scenario will be to put an artificial pacemaker in. Okay, now when we speak about the spread of excitation between the cells, it really starts up in the atria. So the first event is that the atria contract. We've got the right atrium contracting. That's where the SA node is located. It's at the top of the right atrium. And then the right atrium contracts. That electrical activity is propagated to the left atrium, then both atria contract, and then the electrical activity goes down through some of the other conduction fibers that I'll point out. It'll make both ventricles contract. So the first two events are that the atria contract and then the ventricles contract. And I want you to think about why this is important to happen progressively. So why it's important for the atria to fully contract first and then the ventricles contract. Remember, blood is what we're moving here. So we don't want all four chambers contracting at once. It's really important that the atria contract fully first. That's going to squeeze that blood through the AV valves from the atria down into the ventricles. And then when the atria fully relax, then we can see the contraction of the ventricles, which will likewise, again, squeeze that blood from the ventricles out into the corresponding artery. And so it's really important to have the atria fully contract and begin to relax before we see the contraction of the ventricles. Excuse me. And now I mentioned this before, the coordination, the spread of the electrical activity, the conduction, so to speak, is happening via gap junctions. And these gap junctions are one of the structures that are located at the intercalated discs. So the intercalated discs are these junctions between one cell and another. And they've got two important junctions here. One is a desmosome. And a desmosome is a tight junction that helps to anchor two cells together very tightly. It helps to secure that cell-to-cell communication. And it's going to basically resist any of the mechanical stress that we would otherwise experience as these cells relax and contract, continuously over and over. So as the myocardium continuously contracts and relax, we want to really anchor those cells together and prevent any ripping or tearing or shearing and resist any of that mechanical stress. The second structure that we encounter at the intercalated disc at this junction are the gap junctions. And I talked about the importance of the gap junctions, really coupling the electrical activity here. So allowing what's happening in one myocardial cell to be transmitted really quickly to what's happening in another myocardial cell. And that helps to facilitate the synchronous activity of the heart. We also talked about gap junctions as a part of single unit smooth muscle, and it's doing the exact same thing there as well. It's coupling the electrical activity so that the entire muscle acts as one synchronous unit, and so that the electrical activity can be propagated really quickly from cell to cell. So here's an illustration of what's happening between our SA nodal cells. So here's the SA node, that cluster of pacemaker cells. Here are some of the conduction fiber cells, and we can see that the action potential is going from the SA node where it's kind of starting spontaneously down to some of our other cells that are also transmitting this signal out towards the peripheral tissues. And so it's using the gap junctions as a way to send that electrical communication from cell to cell. If we kind of zoom in on what's happening at the intercalated disc, so these squiggly lines here are representing that intercalated disc. And if we zoom in at that area, we'll see those two structures that we mentioned. So here's that desmosome. And as you can see, the desmosome is creating this tight anchoring or this tight junction between these two cells and resisting any type of separating or any type of stress or ripping or tearing of these cells apart. And then the second structure here are those gap junctions. Now I want to point out, because it's sometimes kind of misunderstood or overlooked, the idea that we still have sarcomeres on these cells. So these are still kind of slightly elongated cells the way that muscle cells are. And so we do have that sarcomere, which gives this type of muscle, the striations that we talked about previously. So as the action potential moves from this cell to another cell, it's going to depolarize the second cell. And then what's going to bring about the contraction is really going to be the sarcomeres. All right, let me pause here briefly and see if you have any questions about what we said so far. Okay, So what we're going to look at now is really the steps of conduction, or the conduction system of the heart itself. So from the time that the action potential is initiated in the SA node to the time that both of the ventricles contract and then relax. So the steps are basically, we start off with that action potential in the SA node. Remember, the SA node spontaneously depolarizes and spontaneously fires an action potential, irregardless of any innovation, any neural input, any neurotransmitter input. It can spontaneously open and close those channels and spontaneously bring about an action potential. Then that signal is going to spread throughout the atrial muscle via the interatrial pathways, or the internodal fibers, and then it's going to go to the AV node. After both atria are contracting, then it arrives at the AV node. Now there's a slight delay between the time that it gets to the AV node and then it's transmitted down to the bundle of His. And that is called the AV nodal delay. And the purpose of the AV nodal delay is to allow the atria to fully contract before the signal can go down towards the ventricles. And I mentioned the importance of having that happen so that the atria fully contract before the ventricles begin to contract, so that the blood can move in one direction. Then the electrical impulse gets down to the atrioventricular bundle, then to the, which is also called the bundle of His, that's in between the septum. And then it's going to split off as it gets to the bottom of the septum, and it's going to move to the right and left bundle branches. And then it's going to go up into the Purkinje fibers, and then the ventricles will contract. And this will make a lot more sense as we kind of visually draw this out. So I've got this drawing here that I'm going to walk through. So we start with the SA node. So the SA node is at the top of the right atrium. These cells spontaneously depolarize. This electrical activity spreads throughout the right atrium and then eventually through the left atrium as it gets to the AV node. Now there's a slight pause here in order to allow both of the atria to fully receive that electrical activity and to fully contract. Okay. Then from the AV node, the electrical impulse goes down to the bundle of His. The bundle of His, these conduction fibers are located in between the right and left ventricle. We call this wall the ventricular septum. And then we see that electrical activity kind of split off and go through the right and left bundle branches. This is where the bundle of His fibers starts to split off. And the bundle of His is sometimes called the AV bundle or the atrioventricular bundle. Those terms are synonymous. And then from these right and left splitting of the bundle branches, the impulse will then travel to the Purkinje fibers, which are really at the base of the ventricles. And then from the Purkinje fibers, it's going to allow the ventricles to then fully contract. And the importance of having the impulse go down to the bottom of the heart is so that blood can squeeze up and out of the chamber itself. So we want blood to squeeze down in the atria, and so the electrical activity starts from the top of the atria and makes its way down, so that we have this squeezing of the top of the atria, and then eventually we get the progressive squeezing towards the bottom of the atria. That's going to allow the blood to move in that direction. But when it comes to the ventricles, we want the blood to move up towards the artery, right? Not further down. And so we need the impulse to get down through the septum to the bottom of the heart first, and then it can start spreading out through the bottom of the ventricles and squeeze up towards where we've got the valves. And think about you squeezing a tube of toothpaste from the bottom up. That's going to allow whatever's inside that package to move up and out of the package instead of you squeezing it further down to the bottom. And that's exactly what's happening here. So the electrical impulse starts at the bottom of the heart, and then as the cells there contract first, that blood can progressively squeeze from the bottom all the way up and eventually through the semilunar valves and into the artery. So we see two important concepts that we see here. We see that the atria must fully contract before the ventricles contract. And we see that the contraction of the ventricles must start from the bottom and make its way up towards the valves. Okay. Now the next couple of slides kind of just reiterates all of that. It's a kind of a stepwise sequential process of all of that information that we just talked about. So I'm just gonna kind of go over that quickly. So we start with the SA node, which we said is the pacemaker here. We spread that activity to the left atrium, both atria fully contract. Then it gets to the AV node. Then we get to the AV bundle or the bundle of His, which is in between. So here are the bundle of His fibers in between that that ventricular septum here. Then we get to the right and left bundle branches. So we can see that these fibers kind of split off, some that are going to the right, some that are going to the left, and some that are going to the right here. And then we get to the Purkinje fibers, which are really at the bottom of the ventricles. And as we can see, the distal parts of these fibers extend all the way up to the valves. And so as we squeeze these fibers, as the fibers conduct the electrical activity that allows the contractile cells to squeeze from the bottom up, we then likewise see the movement of the blood leave the chamber from the bottom into that valve.